you watch this video. Not that this material is going to be on it, but I want you to just go ahead and make sure you get that exam finished with, and then come back here. So this week we're going to be starting on the cell and what the different organelles are, and structure and function of both prokaryotes and eukaryotes. So like I said, we'll talk about cells, talk a little bit about microscopy or microscopes, and then we'll look at structures found in both prokaryotes and eukaryotes. So the roles of cells. Cells are fundamental units of life. All organisms are made of cells, and cells can only arise from other pre-existing cells, be it mitosis and meiosis, and we'll talk about both of those later. But this is called cell theory. These are the 3 rules of cell theory. And then all cells share common features. So how do we see cells or microscopy? So we can only use microscopes to view objects and areas of objects that cannot be seen with the naked eye. The first microscopes were used in the late 1500s as a hobby for the rich. Microscopes were used to make scientific discoveries in the mid-1600s. So here you have a very early microscope, and this was what Robert Hooke used. He was the first person to actually see and classify a cell. So you have an oil lamp, because back then, they didn't have electricity. So they had to have lamps going on, and then you can see a little magnifying glass right here. Then you have a specimen holder, you have the objective, and then you have the barrel and the eyepiece right here. So how do we see cells? So there's certain things that you can and can't see with cells. Most of the cells that you've probably had in your high school class, and so a lot of you haven't taken college courses, but you can see most plants and animal cells. You can see very small bacteria. You normally can't see really any of these else, because we normally just can see like these light microscope things, because that's what you have at your disposal. At most universities, a lot of them don't have these electron microscopes unless they're high class, and then most people don't get to use them. But you can see about right here, so you can see a human egg with your eyes. It's a little hard to see, but you can see it nonetheless, but it'll be easier to see with a light microscope. But this is where the human eye can see. This is what you would normally see with a light microscope, and these you would normally need an electron microscope to view. So most cells are between these are micrometers, and 100 micrometers, and can be seen with light microscopes. Smaller things like cell organelles, viruses, and individual molecules need electron microscopes. So getting into the difference between prokaryotic and eukaryotic cells. So in your outline notes, you have these cells on your paper, so you can label as you go. You can use that for studying, but you also have these in your textbook. You can go to page 97 to 101 in your textbook if you don't print these out or you don't want to label them or anything like that. So there are 2 main types of cells. You have prokaryotes, which this is a prokaryote up here, and then you have eukaryotes down here. All cells have a common structure and function. So we have the plasma membrane, which is a flexible outer boundary. So you can see right there is the plasma membrane. Cytosol, a lot of times it's called cytoplasm, which is just the jelly-like substance on the inside. Both cell types also share flagella. Not all eukaryotic cells will have a flagella, just like most cells in the human body do not have one. But some of them do, kind of like sperm cells, they have that flagella. And most bacteria will have flagella or some kind of flagella. And it's just a long, thin extension of the plasma membrane that helps with moving in fluids. So right there. And then we have ribosomes. Both prokaryotes and eukaryotes will have ribosomes. This is how we synthesize protein, and this is where you synthesize protein. In prokaryotes, these are free floating in the cytoplasm or the cytosol. That's what these little dots are out here. In eukaryotes, they are connected to the endoplasmic reticulum, the rough endoplasmic reticulum along the outside of the nucleus. So some structures that are unique to prokaryotes. Prokaryotes are organisms that include archaea and bacteria, and they do not have a nucleus. That is the main difference between prokaryotes and eukaryotes, is there is no nucleus. In prokaryotes, the DNA is housed in a region called the nucleoid. So it's just the center. The DNA is all housed right here. And then there is the cytoplasm. So prokaryote cells have structures that eukaryotes don't. Where are you not moving? There we go. On the outside of the plasma membrane, prokaryotes have a cell wall. Plant cells and fungus have cell walls. Animal cells do not, but it says that all prokaryotes don't. All eukaryotes don't. Most prokaryotes do have a cell wall, which is just a very rigid structure outside of the plasma membrane to give it a little bit more structure. Then you have the glycocalyx, which is an outer covering on many prokaryotes. That's just a slimy, sticky layer that kind of allows them to attach to other organisms or to surfaces. So eukaryotes are a little bit more complex than prokaryotes. Prokaryotes really just have that cell wall, ribosomes, and DNA, which is housed in the center of the cell. But eukaryotes do have a lot more organelles, and so we're going to get into that right now. So the very special thing about eukaryotes is that we have the addition of the nucleus, which is where the DNA is housed. Now, there are DNA in chloroplasts and mitochondria, but majority of your DNA is found in the nucleus. So there's your nucleus right there. It's real big, circular. It's not always in the center. A lot of times it's kind of off to the side a little bit, but it is a very dark space that you will see on the cell. And when you look in a microscope or a light microscope, you'll see a big dark area. So chromatin or DNA structures found in the nucleus. The chromatin is just the long strands of DNA. The nucleolus is in the middle of the nucleus, and that is where we store ribosomes, and that's where they're made before they get shipped out to the rough endoplasmic reticulum. The nuclear envelope is a membrane that surrounds the nucleus to protect it. So right there, it's the one with all the holes on the side of it. It has two membranes. It has an inner and an outer membrane. And the nuclear pores, which are those holes I just talked about a second ago, and those regulate mRNA, or just RNA in general, coming in and out of the nucleus. So there's a nuclear pore. Eukaryotic cells often work together to form tissue and need to hold certain shapes. So they tend to have a cytoskeleton, which is just an elaborate network of filaments. We normally call them microfilaments. And they just run through the cytosol, so you can kind of see some structure going on right here underneath all of this mess. And it just keeps the cell together, kind of like a skeleton. So the cytoskeleton is made up of microfilaments, like I said, and these are large fibers, solid rods that act as a cell shape. A lot of times we call this actin. So you can see right here, we have the microfilaments right there. Intermediate filaments, or we can also call that keratin, are medium fibers, but they're not really big, not really small, so we call them intermediate filaments. Those allow you to bear tension, so you got right there. And then microtubules, which we also call tubulin. Those are very small fibers which help support the cell and form a highway network for motor neurons to pull cargo around. So these microtubules are also what help in cell division, but we'll get to that in a couple months, or not a couple months, a couple weeks whenever we get into that. But these just help pull organelles around, or anything else that needs to be pulled from one side to another. So microtubules also make the centrioles, which are these things right here. And like I said, that's what helps split the cell apart or move the chromosomes apart during mitosis. That's a centriole right there. It looks like two pieces of pasta, so like those open, I just forgot what they're called, like ziti noodles, kind of, like got like the open edges. That's what it's talking about, two pieces of pasta kind of stuck together right there. That's what the centrioles look like. Like I said, we'll talk more about those when we get into mitosis. All right, so mitochondria, you may have heard, is considered the powerhouse of the cell, and that is because this is where energy is made. It's found in almost all eukaryotic cells. So even though plants go through photosynthesis, they also go through cellular respiration, and they have these mitochondria in there. Now, don't ever get confused, because when we talk about mitochondria, we're talking about multiple, but mitochondrion is talking about one single organelle. So mitochondria is multiple, mitochondrion is just one. So again, like I said, the powerhouse of the cell. It makes ATP, which is what we consider chemical energy, by extracting energy from sugar, fats, and other fuels. And as I say, we'll get into this more next week and how it does this. But they do have two membranes. They have an inner and an outer membrane. I thought I was going to show you, but it's not. So they have this outer membrane right here, and then this inner membrane does close up all the way, but that is the inner membrane. And they have these cristae, which are just the folds. Okay, but mitochondria and chloroplasts both are very similar to prokaryotes. And like I said a couple of slides back, they do have their own DNA, both mitochondria and chloroplasts. So it is thought through this endosymbiotic theory that early eukaryotes engulfed this prokaryotic cell. A mitochondria and a chloroplast were considered to be once free-living prokaryotic cells. So a eukaryotic cell swallowed them whole and was able to form cells that could do both cellular respiration and photosynthesis. And eventually, the eukaryotic host cell developed a symbiotic relationship with the prokaryotes, just saying that both of them benefit. So this mitochondria is able to get everything it needs, the proteins, housing everything it needs, and in return, it gives the eukaryotic cell energy. So they both benefit from this relationship. There's many structural differences between mitochondria and bacteria. Similarities, excuse me. There's many structural similarities between mitochondria and bacteria that support this theory. So both of them will have two membranes. All of them have DNA, circular DNA, and ribosomes. And they have their own organelles. That means that they can grow and reproduce. So one cell can have thousands of mitochondria. Therefore, higher metabolic activity. So, now it's going to go into the parts. So we got the parts of mitochondria. You have the outer membrane the inner membrane with these folds called cristae. On the inside of the cristae, you have a cytosol-like fluid, but when it's in the mitochondria, we call it the matrix. It's just like the cytoplasm or the cytosol. It's just a jelly-like substance that's in between these cristae, but we call it matrix. Right there. So ribosomes make proteins. They do this by reading that mRNA. Like I said, we'll get into this later when we talk about protein synthesis. But the DNA makes a code of mRNA. The mRNA comes out to a ribosome, and then the protein is made. So you can see these brown dots. You have free ribosomes that are all out here, and then the rough endoplasmic reticulum has a bunch of them on it. They do not have a membrane, and they're made up of protein and RNA. So free ribosomes, they're free floating in the cytoplasm. Here you can see a ribosome. You have a smaller or larger subunit with a smaller subunit. Then you have some that are attached to the endoplasmic reticulum. So the endoplasmic reticulum, we also call it the ER, consists of parallel interconnected cisterns. So like I called the cisternae in the mitochondria, so you have these folds. The ER has it as well. So it kind of looks like a big stack of pancakes going on. It's a continuation of the nuclear membrane. So they're not really like separated. They're really just a continuation of the nuclear membrane. And there's two types. So we have the rough endoplasmic reticulum, which is right here with all the dots on it. And then you have the smooth endoplasmic reticulum over here. So the rough, the reason why they call it the rough is because it has all those attached ribosomes. And so under a microscope, until they could get really accurate images, it looked kind of like sandpaper. So they called it the rough endoplasmic reticulum. And then So functions. It makes proteins. That's why we have all these embedded ribosomes on it. It helps modify proteins. So it can add carbohydrates to folded proteins. And it helps with exportation. So it is the first line of defense whenever we're trying to send proteins out of the nucleus. And we'll talk about that in the next coming weeks. But it is the first line of defense to sending out proteins. All right, now I have the smooth ER, which is this over here. So it is a continuation of the rough ER. The smooth ER, because it doesn't have those ribosomes, it doesn't have that sandpapery-like image. So that's why we call it the smooth ER. So it helps with metabolic processes like lipid synthesis, calcium storage, breakdown of carbohydrates into sugar. It really just helps kind of clean everything out. So in your lipid cells, they actually have a lot more smooth ER than normal cells. All right, moving on, we have the Golgi, which is this guy over here. It helps package things up as well, and it does have the cisternae too, just like pretty much everything else. Looks like a big old stack of pancakes. And the Golgi, you can really just consider it like your post office or FedEx. So it's going to get vesicles from all over the cell to it, and it's going to decide where it needs to go and ship it out. So like I said, modifies, concentrates, sorts, and packs proteins and lipids received from the ER. So it will come from the ER into the Golgi, go through the Golgi, and then the Golgi will tell it where it needs to go from there. So, just like I said a slide ago, Golgi receives and sends molecules all across the cell like USPS and enzyme. It occurs in stages, so this is the ER back here at the end. So it actually goes through pretty much all of these cisternae. We call it a cisternal maturation model. So they come in this back, kind of engulf in all of these cisternae coming in, and then they come out in what we call vesicles. So they'll come out, have proteins all in them, and head to the cell membrane. So they'll start at the cis end, which is the receiving end, and come to the trans end, which is the shipping phase. Is this going to be a video? Nope. Okay, so here's the Golgi, here's the cis end, so this is the receiving end. So, and then this would be the ER. Yeah, so this is the ER. This is shipping vesicles with proteins in it. They will go in, go all throughout this process, and then come out the other end, and you have the vesicles going. This one's starting to go. Vesicles going where they need to go.
Coloring one of the compartments will help illustrate the cisternal maturational model.
Vesicles fuse with the cis-Golgi network
In this
until it
model,
begins to mature
vesicles
and move into the
fuse
Golgi
with the
stack.
cis-Golgi
And then the
network
ergic
until it
vesicles create a
begins
new
to
compartment in its
mature
place.
and move into the Golgi
Compartments
stack.
with their proteins
And then the
mature to become
ergic
the medial.
vesicles create a new compartment in its place. The compartments with their proteins mature to become the medial and then the trans compartments of the Golgi stack, where further protein modifications take
gradually
place.
mature and progressively move through
The
the Golgi in
compartments
the
of the
cis-to-trans
Golgi
direction,
gradually
carrying their
mature
contents
and
with them.
progressively move through the
The
Golgi
trans-Golgi
in the
network acts as a
cis-to-trans
sorting and
direction,
distribution center,
carrying their
directing molecules
contents
to their
with
final
them.
destination.
The trans-Golgi network acts as a sorting and distribution center, directing molecules to their final destinations.
The proteins leave in
The
vesicles,
proteins
which bud off
lead
until
in
this final Golgi
vesicles,
compartment
which bud
disappears
off
and
until
is
this
replaced by a
final
maturing
Golgi compartment
immediately behind it in the
disappears
stack.
and is replaced by a maturing compartment immediately behind it in the stack.
All right. So that's all just what I explained. You can also see some of them going medial. So it could be proteins that just the Golgi needed. So it could come in here and be like, oh, I need proteins over here, so it can go medial and back in throughout the Golgi stacks. So how does the Golgi control where to go? So think of the UPS. Earth Postal Service uses zip codes to tell carriers where to take the letters. Vesicles from Golgi are tagged with molecules like phosphate groups, and that is how the Golgi knows exactly where it needs to go. So either it needs to be exported to go into the plasma membrane, or it needs to be destroyed and go to a lysosome. And we'll talk about lysosomes in just a second. Now we're going to talk about lysosomes. Are digestive compartments that break down molecules. I always say think of lysosomes like the trash pickup people. So they're going to come up and they're all going to break down all of the garbage in our cell. So they contain an enzyme that hydrolyzes large molecules, so just going to break them down. And so you can see the lysosome right there. Arises from budding off of the Golgi apparatus. Enzymes are made in the rough ER. So the ER will make that enzyme that it needs. It will come to the Golgi, and then it'll be like, oh, okay, well, I'm going to be a lysosome. And it'll bud off, and it'll have those enzymes that were made. Because remember, last week we talked about enzymes are a type of protein. So you can see this engulfing it. So you can see there's something that it's trying to get rid of. The lysosome literally like engulfs it. So it just wraps around it. And it'll start to degrade those materials, break them down, and it will reuse them. So we don't want to waste anything. It will break it down and use what it can again. Okay, I can also, so that's called phagocytosis. So it's taking in this food or organisms, whatever it needs to do. Lysosomes help by blood cells in fighting off pathogens. So the lysosome in the white blood cells is actually what helps break down those pathogens or infection-causing bacteria. You have your peroxisomes, which a lot of times people don't really talk about them, but they're organelles that have enzymes to remove hydrogen atoms from substances and transfer them to O2. And you got those little peroxisomes right there in blue. and they help create hydrogen peroxide, which can neutralize toxins. All right, now we're moving on to the difference between plant cells and animal cells. You should know this from high school or from your high school biology course, but just kind of a brief overview. So this is in page 97 to 101. This is a plant cell, so you have the plant cell in your book, but it should also be in your note outline for you to go through it on your own as well. So plant and animal cells share many organelles together. There are a couple of differences. So while they do have a plasma membrane, and they have cytosol, and they have a cytoskeleton and a nucleus, and the smooth and rough ER, they have the Golgi, they have the peroxisomes, the mitochondrion. They have all the same things, but plant cells tend to have a couple more things. So they have a cell wall. Animal cells do not have cell walls. Other eukaryotes will, so fungus do. Majority of your protists do have cell walls. And so animal cells do not have cell walls. Come on. All right, so just an outer layer in plants. It's made of cellulose, and it just helps protect the cell and helps it maintain its shape. There you go. And it contains a channel called the plasmodesmata, which connects the cytoplasm of adjacent cells. So you have all of these holes, and it just connects it to the cells that are right next to it. The next cell will have these holes as well, so it's just openings where we can exchange information in between. And this more or less is true. Animal cells have vacuoles too. They just tend to be smaller when we have them. But vacuoles just hold on and store molecules for later on. Plants, it's more appropriate to say they have a large central vacuole. So they'll have a really big one. Most of the time it just helps store sugar that's not ready to use yet, or water, or anything like that. It'll just hold on to it in that large central vacuole. So it's filled with fluid called cell sap that houses inorganic molecules as well, so potassium and chloride. And it absorbs water to enlarge when the cell is growing, so it doesn't have to make new cytoplasm. So if the cell gets bigger, we can fill this up with water so that we don't have to make any more cytoplasm. Also a plant thing, they have chloroplasts, which help them with photosynthesis. So that's what converts sunlight into chemical energy, or ATP. But still, you can see they have these mitochondria in their cells as well, but they have the chloroplast. There's no other eukaryotes that have this, except for algae. We don't consider those plants anymore. They're considered a protist. There's some protists that have chloroplasts, but fungus do not have chloroplast, and animal cells do not have chloroplast either. We'll talk more about photosynthesis, not next week, but the week after, I believe. And then we have the endosymbiotic theory, like I stated earlier, that these chloroplasts were once free living organisms, and they were just engulfed by a eukaryotic cell. So getting into the parts of the chloroplast, we'll get more into this later when we talk about photosynthesis. But you have the outer layer. This is a two-part membrane. So you have this outer layer, and then you have the inner layer. And then you have these things. We have these stacks. This whole stack is called a granum. Each individual plate, I guess you can call them, are called thylakoids. And in between, just like when we have the mitochondria, you have the space in between it. But when we call the space in a chloroplast, it is called the stroma. Now we're moving on to the cell membrane. So, as I stated earlier, we have a fluid mosaic model. So even though we do have these phospholipids, everything is able to move. It's all fluid. It's able to get around. It's never really set. So this functions in providing a site for energy reactions, nutrient processing, and synthesis. The cell membrane is selectively permeable, so not everything can get into it. It's very selective about the type of molecules it lets in and out. All right, so we talked about phospholipids in the last chapter, but just to remind you, you have two phospholipids. They're both facing away from each other. The hydrophilic part is on the outside edges, while the hydrophobic is on the center part. So cell surface membranes can carry out several functions. So we have for transport, enzymic activity, cell transduction, cell-to-cell recognition, intercellular joining, and the attachment to the cytoplasm or the extracellular space. So here's just what those look like. So we have proteins that help with transport, help molecules come in and out. And we'll talk more about this later. And then we have enzymic activity. So we have enzymes that will pop off here and create or catalyze reactions. We have signal transduction. So something will come on here and send a signal, and this second messenger will go off and tell its friends what to do. Cell-to-cell recognition, so we'll know what each other are. Intercellular joining, so that the cells connect to each other. And then attachment to the cytoplasm or the extracellular matrix. So here is, looks like actin and myosin right here, and they're connecting together. And then you have a microfilament down here that's connecting. So we have proteins that help with that as well. Getting into cell transport. So a cell must exchange molecules with the outside world, but some of these molecules may be too big or polar and cannot make their way through the hydrophobic section of the cell membrane. So in order to do that, we have lots of different ways to come up with that. So you have passive transport and active transport. Passive, it uses a concentration gradient to move molecules through the membrane. We have different types, and we'll get into each individual one. But active uses energy to push molecules through the membrane. So passive does not use energy, while active does. So getting into this is just examples, but getting into passive right here, we have diffusion, and you're going down a concentration gradient. So there is a ton up here and not any in here. So these molecules are getting too tightly compact, and they want to move away. A lot of times diffusion is only with gas molecules because they're the only thing that can really get through. And then you have facilitated diffusion. We have a channel protein right here where these molecules are able to go down their concentration gradient just with this opening. And then you have other transporter molecules which will only allow one or two at a time. So you'll have it open to the outside up here. A molecule will come in there, and then it'll open to the inside of the cell, and this molecule can leave. And then active transport, this is a sodium-potassium pump right here, which we'll get into probably in a little bit. But ATP is that energy, and it causes, just like over here, we have this, but this requires energy to open and close this pump. So getting into it, like I said, diffusion is small molecules that move down their concentration gradient. So they'll go from a high concentration to a low concentration. So you see right here, there's a bunch up here, none on the inside of the cell. And so these organisms are not organisms, but these molecules are going to start going through the cell membrane to make it equal on both sides. And that's what we want. We want it equal. Facilitated diffusion is when you use a channel protein or a carrier protein. And this is what glucose has to use. So glucose has its own special channel protein to get in and out. So does water, but we'll talk about that on the next slide with osmosis. So this is just for large or polar molecules, because this section right here is nonpolar, and so water-based molecules are not able to get in through here. Neither are large molecules, so they have special proteins to get them in and out. Here is osmosis, like I talked about. So it's just like facilitated diffusion, you have your own little proteins, but the protein that allows water to come in and out is called aquaporin. And so you can see right here we have the water, and because there's a higher concentration of solute over here, the water wants to balance that out, and so the water will move, causing the water to go up over here. These are very important terms to remember when it comes to water and molecule balance. So when we talk about hypotonic, that means that the inside of the cell had a lower concentration of water, and so the water floods into the cell. If the water makes the cell too large and it's not able to compensate by building on more cell membrane in time, it will pop, and this is called lyse. Plant cells, they don't really pop because of that cell wall. They just become turrig. So they just become really full. Isotonic is normal. So water's coming in and out at the same rate. We have an equal going on, so it's called isotonic. In a plant cell, that is called flaccid. Everything is just okay. When it's hypertonic, the concentration of water was higher on the inside, so it wants to come out. So water keeps leaving the cell, and it becomes shriveled. In plant cells, we call that plasmolysis. And I wanted to bring up here, this is why it's important why you don't hydrate very quickly after you've been sweating a lot. Because then if you hydrate faster than your cells can handle it, these cells can pop. Now, it takes a lot of water. I think people who have died from this kind of thing is taking several gallons of water in a very short amount of time. But they die because their blood cells are popping. And not just blood cells, it can be any cell. They are popping because the concentration of water is so much higher outside. So the water wants to go into the cells, and then they do this popping sensation. All right, so getting into active, there's several different types of active. So we have endocytosis, and then there's several different types inside of that. So we have phagocytosis, is when you bring in food or very large molecules into the cell. Penocytosis is normally like water or gases or smaller molecules coming into the cell, and we're creating a vesicle with those inside. Or receptor-mediated endocytosis, which is where we're just engulfing receptors. Right, and then exocytosis is the exact opposite. So that is when molecules are leaving the cell. And I thought this was supposed to be, maybe not. I thought it was supposed to be an interactive thing. But in exocytosis, the vesicles go up to the plasma membrane and they fuse with it, and then allow the molecules that they have inside of this vesicle to be released into the outside of the cell. All right, another type of active transport that you have in your body that's very, very important is the sodium-potassium pump. So what happens is we are trying to get potassium out of sodium out of the cell, potassium into the cell. So this cytoplasmic sodium combines, you get 3 cytoplasmic sodiums to combine to it. We have ATP. So energy is used to close this off and open up to the outside. So then these 3 sodiums leave. Once those 3 sodiums leave, these extracellulars on the outside, potassiums are able to connect in. We have another energy being expelled so that we open up back into the cell, and those potassiums can leave, and it'll keep doing this over and over again. All right, so this week you have to do the exam, which hopefully you've already completed. You have chapters 6 and 7 reading quiz. You also have the cells assignment, which I don't think is very long. I think it was only like 20 to 25 minutes worth of stuff. But next week we'll talk about metabolism, so we'll talk about cellular respiration and photosynthesis and all that stuff. So I'll see you back here next week.